So this lecture is our first dive into the chemical senses. So we're going to focus on taste to start with. Um, so big things about chemical senses: animals depend upon them for recognition of nourishment, poison, and potential mates. They're some of the oldest, most common sensory systems among all species. Again, reminder, dolphins are the only ones that have a sense of smell, but we'll get to that later. They do have a sense of taste. They do have some chemical sensation. The two that we're going to, the three main chemical sensations are taste, which is gustation, olfaction, which is smell, and chemosensation, which is like detecting irritants on the skin. So those are three major forms of chemical senses. And in taste, of course, you kind of all know this already. We have our five major tastes that we have. We have salty, sour, sweet, bitter, and umami, or savory. And we're going to talk about how those signals come about, how they're elicited from the tongue to the brain. So, a couple quick interesting facts first, though. Bitter does tend to be a detector for poisonous foods, so there's a reason that people tend to shy away from really bitter things, is because it tends to mark something that's not good for you to eat. It might be poisonous. Bitter can be detected in its tiniest of ranges, where down to the nanomolar range of concentration, so we're extremely sensitive to it. And then also artificial sugars such as aspartame and saccharin are about 10,000 to 100,000 times sweeter than sucrose. So the concentration needed to achieve the same level of sweetness is like 1/10,000th or 1/100,000th of natural sugars. So we have taste sensation. It comes down to like, what is flavor though? And what we're going to find out is that flavor is a combination of multiple sensory modalities, meaning we have different senses that come together and actually give us a cognitive understanding of what flavor is. So it's about a combination of different taste receptors that are activated, the combination of different odor receptors that are activated, the texture, the touch of the food, the temperature of the food, and maybe even some pain as well. So in those nachos, you might have some jalapenos with capsaicin in it. So that's going to elicit a pain response in addition. So all of these five things are going to go into what we perceive as flavor. But let's break down what taste is all about. So the organs of taste. Okay, you basically have your tongue, your pharynx, your epiglottis, palate, nasal cavity, basic structures you all kind of know from other biology classes, understanding like what the major parts are. So you might have seen a diagram like this before about areas of sensitivity in the tongue. The tip of the tongue tends to be more sensitive to sweet, sides to salty and sour, and the back of the tongue to bitter and umami. Now, this doesn't mean that that's the only place that you actually sense those tastes. It's just that's where you're more highly sensitive to it. Taste is spread throughout, and we'll kind of see that through some of the recordings we take a look at. So here's the basic setup of the structural components of the tongue. We have the valate, the foliate, and the fungiform papillae, okay, which are spread throughout. You can see the fungiform are kind of these dots near the front up here. The foliate is this ridge-like thing over here on the side, and the valate is back here. Okay, so those are the different papillae, and you can kind of see it blown up in the second picture, where you have the papilla right here. Okay, and in that papilla are these little things right here, and these little dots, these are your taste buds. Those little dots right there, those are your taste buds. Now what's important there too is you have these little taste pores on the taste buds, and that's what's going to gain access to that area in that groove where food is going to get caught and you're going to be able to sense taste. There are little microvilli, basically meaning like little things sticking out. They're going to catch some of that food, okay, and bring some of those taste chemicals into the taste cells, which are right here. Those are the things that are going to expose themselves to the taste pore. So each of the taste buds has a given number of taste cells associated with it. And you're also going to see these things called basal cells back here. They're important because they're actually going to replace the taste cells. They kind of set a structure, but they're kind of like stem cells for the taste cells. So eventually they'll become one of those taste cells after a while. And then everything's going to go out via these gustatory afferent axons. So there's a synapse between the taste cell and the axon. So the taste cells don't actually have axons themselves. They just kind of release something onto that afferent axon, and it sends it out to the rest of the body, right to the brain. So what happens electrically in a taste cell. So here we have 3 different taste cells, 1, 2, 3. They're exposed to 4 different taste chemicals. So you have your salty sodium chloride, you have bitter quinine, acetic hydrochloric acid for sour, and then you have sweet for sucrose. And you can see cell 1 actually does respond to 2. It responds to the salt and it responds to the acid. Cell 2 actually responds to the salt and the acid pretty strongly for both, and even also responds a little bit for bitter. And cell 3 only seems to respond to sucrose. So the taste cells are not specific to a given taste. But notice they also don't generate action potentials. You're only generating a membrane depolarization, which in the end is going to cause action potentials in the axons that are leaving. And here you can see in the axons, anytime there was a depolarization, okay, you can see an increase in frequency of action potentials. So they're firing constantly these action potentials, but they're going to increase their frequency when that taste receptor cell that's associated with that axon becomes activated by whatever taste molecules bind to it. So a couple interesting tongue facts. You have about 2,000 to 4,000 taste buds in your tongue. Catfish have the most of any animal. They actually have about 20,000 taste buds. They actually have them all over their body. You have some in other places too. They are not just in your tongue. You have taste buds in other locations, but catfish has almost 20,000 taste buds. The basal cells do replace the taste cells about every 2 weeks. And if the nerve is cut to the tongue, that corresponding taste cell that goes with that axon is going to degenerate. Okay, so great, we've got the taste cells, they're getting a signal coming, something's binding to them, it's generating a signal, and we see what the output is and how it affects action potentials, but how does it actually happen at the taste bud itself, at that taste cell, to generate those depolarizations? Well, it can happen one of 3 ways. One, it can directly, that tastant can directly flow through an ion channel. Second one, it can block the flow of ions through a channel, or we can activate G protein coupled receptors. So 3 different mechanisms of transducing the extracellular signal into that depolarization that's going to cause the exponential change in the axon. So salty is our first one. Salt sensitive taste cells have a special sodium selective channel, okay, right here. It's a millerite sensitive sodium channel, okay, and basically it allows sodium to flow into the cell. You can start to detect at about 10 millimolar sodium in the external concentration. Right? So as sodium comes in, the membrane starts to get depolarized, and that's going to lead to voltage-gated sodium and calcium channels opening up and causing more depolarization, which again is going to cause our vesicle release and give us the action potential increase in the afferent axon that's coming out. Okay? So sodium is pretty straightforward, and it kind of makes sense. You increase external sodium concentration, it's going to go into the cell, and you're going to get an increase in depolarization of the cell that way. Sour also flows through the exact same through the millerite sensitive sodium channels, okay, except it's hydrogen ions. Since we're talking about an acid, we're talking about an increase in hydrogen ions, so they're going to start flowing through there. The other thing they're going to do is stop potassium from flowing out of the cell. They're going to block it. So if you block potassium from flowing out of the cell, which would tend to hyperpolarize the cell, you're going to get a stronger depolarization. So we're going to get, again, another depolarization in the membrane, again causing calcium to come into the cell, and again causing a vesicle release. So these channels actually act as a pH sensor. They're going to detect increases in H+ concentration. Right, as the hydrogen ion concentration goes up, more of it's going to come in through that sodium channel and block that potassium channel, depolarizing the membrane more. So anytime you have one of these receptors, that's what's going to happen. Bitter is going to work completely different. Bittersweet and umami are going to work completely differently. There's going to be a G protein coupled receptor for it. Okay, so G protein coupled receptor binds bitter, sweet, or umami. It's going to activate phospholipase C, which in turn activates IP3, which the main thing here we're going to get out of it, again, is internal calcium depolarizing the cell, but it's going to come from calcium inside the cell. This calcium right here, the calcium in stores, doesn't count towards the concentration of calcium inside the cell because it is inside a membrane. Since it's inside a membrane inside the cell, it's actually isolated from the intracellular space. But we can release it, and IP3 does that. So again, calcium comes out of that store, depolarizes the cell, more calcium comes into the cell, and synaptic vesicles get released. So there's about 30 members of the bitter receptor family that have been discovered so far. So lots of different things combined with bitter. The other thing too that's kind of interesting is you can't actually distinguish between different types of bitter. So you just basically know it's bitter or it's not. So some interesting things about bitter there. Sweet and umami, a little bit different. Sweet and umami are both going to use a dimer of 2 receptors for their detection. So both of them contain the T1R3 dimer receptor, but they're going to partner with a different one. We have T1R1 and T1R2 are different partners. Okay, T1R2 for sweet and T1R1 for umami. And they're going to use basically the same mechanism we saw with bitter, except now we have to have both these receptors activated. Now, in your papers, you will find that you're researching, or at least the groups that do sweet and umami, you're going to see that one receptor is enough to give a small signal, but to get the full strength signal, you're going to need both receptors to really isolate out, hey, this is sweet, hey, this is umami flavor. Okay, cool. So now we've got those axons leaving the tongue. They're going to go up to the brain. And we're not going to talk about all the processing in the brain, but we want to talk about the pathway where they're going to go. So everything's going to leave here, leave the tongue, and things are going to make their way into the brainstem, okay, through some of the cranial nerves. It's not going to go through the spinal cord. Okay, it bypasses the spinal cord, goes straight into the brainstem. Okay, and then once it gets to the brainstem, the real key here is it's going to go through this area right here. It's going to go through the thalamus. So it's got that thalamus as its relay station. So incoming signals all go up here into the thalamus, and then they're going to get processed out in the gustatory cortex, which is over here. That's in the parietal cortex, so out in the parietal lobe. So understanding that before it gets that parietal lobe for further processing, it's got to go through that initial relay stage of the thalamus. So some things about neural coding of taste. The original hypothesis was that we had labeled line coding, which means there was individual taste receptors for each stimulus coming in. Things were very specific. Taste cells were very specific for sweet, salty, sour, bitter, umami. But what they eventually found out through a lot of recordings and measuring is that it's more about what's called population coding. It's patterns of activity through broadly tuned neurons partnered with other modalities are going to lead to us what our actual taste is. So population coding is basically saying that, okay, if A is active to this level and B is active to this level and C is not active at all, I know that that's a certain type of sour. Okay. Whereas if A is barely active, B is really active, and C is sort of active, well, that's going to be a bitter flavor. So it's understanding the combinations of activity, because things aren't extremely specific, that's going to lead to this thing called population coding.